it is a great pleasure for me to announce, on behalf of the European Association for the Study of Obesity, the 2021 winner of the Friedrich Wasserman Award, Professor Hans Hohner. He studied medicine at the universities of Regensburg and the Technical University of Munich. After a postdoc phase at the Institute of Biochemistry, Microbiology and Genetics of the University of Regensburg, as well as the Department of Internal Medicine, Division of Endocrinology, Metabolism and Nephrology at the University of Ulm, Hans Hohner became deputy head of the clinical department of the German Diabetes Center at the Heinrich Heine University Düsseldorf. Since 2003, he is a chair and director of the Else Kröner Fresenius Center for Nutritional Medicine of the Technical University of Munich. His institute continuously grows at two locations, Freising and Munich, and is now the most recognized and well-known nutrition center in Germany, focusing on obesity research. Hans Hohner is also an elected member of the National Academy of Science, the oldest research academy in Germany, and he served as spokesman of the German Competence Network for Obesity. Hans Hohner is a pioneer of national and international obesity research, especially in biology of adipose tissue. In 1987, he described for the first time the in vitro differentiation of human adipocyte precursors and refined it for the applicability in subsequent original research articles. This human adipocyte model is still the preferred model for human studies. He initiated and published numerous articles on the secretory and inflammatory function of human adipocytes, including leptin and other cytokines. This excellent work was extended by the functional characterization of genetic risk loci for obesity and diabetes, which has been very successful and resulted in high-impact papers. He contributed to the development of new bioinformatics and experimental approaches to identify obesity-associated single nucleotide polymorphism and to elucidate their molecular mechanism. Besides these activities in basic research, Hans Hohner was principal investigator of many clinical studies focusing on weight management and other intervention studies, including behavior and pharmacotherapy trials. Acknowledging the complete list of Hans Hohner's scientific achievement is certainly beyond this presentation, but his research is best reflected by more than 450 articles listed in PubMed. Among those articles are landmark studies which have been published in the highest ranked international journals and are frequently cited. The whole career of Hans Hohner has been characterized by outstanding honorary engagement for professional organization. He was the secretary of EASO as well as the president of the German Obesity Society. Since 2014, Hans Hohner is editor-in-chief of the EASO journal Obesity Facts. He was also editor-in-chief of the German obesity journal Adipositas and chair of the Guidelines Committee of the German Obesity Society. Moreover, thanks to the enthusiasm and efforts of Hans Hohner, there has been an increase for awareness for obesity in the lay public and among politicians. Owing to his work, we're expecting a dedicated disease management program for obesity in Germany in the coming year. He also published a German standard educational book on obesity and contributed to many book chapters. As a member of the executive board of the German Nutrition Society and chair of the German Diabetes Foundation, he was an ambassador for obesity-related topics, reaching far out to other disciplines. His advice is highly regarded in the media, and Hans Hohner is a sought-after expert for all topics on obesity and its comorbidities in TV shows, news, broadcasts, and newspapers. The German Obesity Society considered Hans Hohner as one of the fathers of obesity research in Germany. Therefore, he received the first award for lifetime achievements in the field of obesity from their society. In summary, the Executive Committee of EASO is delighted to award Hans Hohner the 2021 Friedrich Wasserman Award because we believe that he has shaped obesity research in Europe and deserves the award for his lifelong achievements. I give now the floor to Professor Hans Horner, who will present his famous work.
Thank you, Natalie, for this very kind introduction. And this just tells me how old I have become, but it's, I think, a good opportunity to look back and to show what we have done over almost 40 years of research in the fat cell. This is the title of my brief lecture. It's a brief journey from the fat cell in the early 80s up to today, and I hope you will see that a lot has happened in the field of obesity, and I would like to share this with you. So the starting point was that in the early 80s, little was known about the origin and the function of adipose tissue in general. It was particularly unclear if human fat cells can be formed after early childhood and if precursor cells exist at all. This was in contrast to data from rats and mice. However, in our first attempts, we wanted to establish a human system to look at fat cell development, and it didn't work if we used some mouse media. And over the years, we found out that the presence of fetal calf serum in these culture models was inhibitory for fat cell differentiation in humans. And so we put a lot of efforts to really identify the factors which are promoting differentiation in human adipocyte precursor cells, and also studied factors which are inhibitory. And this is briefly summarized on this slide. So there are precursor cells in humans as well at every age up to the age of 80. This is now for sure. In the 90s and later, a lot of studies have been done on adipose tissue, and this shows a complex picture of adipose tissue biology and function, and it illustrates a very adaptive, complex system which includes a variety of cells and a variety of functions. And the central thing here is that fat cells have been identified to produce numerous factors which are released into the circulation and to the environment and may exert a lot of functions. We were particularly interested in the role of enlarged fat cells because there was already evidence that large fat cells are associated with comorbidities. And we then tried to fractionate fat cells. First, you see here the association between BMI and mean fat cell volume. And what we wanted to do is we wanted to separate fat cells according to volume or density. This was done by Thomas Kurk, and he established a system to separate the cell fraction into 4 classes as shown here. And we did a lot of studies to look at adipokine secretion, ER stress, mitochondrial function, and insulin action. And in all these studies, it turned out that enlarged fat cells are much less responsive and have a disturbed metabolism. Just one example for this, you see here the 4 cell fractions, and the right one is a fraction of the large fat cells, and you can easily see here that factors such as IL-6 or IL-8 are mainly produced by these enlarged fat cells, illustrating that fat cell enlargement means inflammation and insulin resistance. If we summarize this, we could also find evidence that these secretion of products is associated and linked with the clinical consequences of obesity. And this is from a review paper illustrating which products are associated with which clinical consequences. And so there is a clear link between adipose tissue function and the clinical comorbidities of obesity. I will not go into detail. I think most is well known. We were also interested in the role of fatty acids here, and in 2004, Gerard Ayot, one of our collaborators and friends, developed this hypothesis claiming that the ratio between omega-6 and omega-3 fatty acids is important for the early determination of the adipose tissue mass. This was based on studies from rodents. There was circumstantial evidence from human studies. For example, on the right-hand side, the ratio between omega-6 and omega-3 fatty acids was increasing in breast milk, and it was postulated that this could be a cause for childhood obesity. And as a trained clinician, I was always interested to do this type of translational research, and so we studied this in a proof of concept human intervention study, the so-called Infect Trial. And the idea was, if we reduce this omega-6, omega-3 fatty acid ratio in the maternal diet during pregnancy and lactation, this could be a step for a kind of primary prevention of childhood obesity. This is a study design. It was a two-arm trial, and pregnant women were recruited before the 15th week of gestation. The intervention was supplementation of 1.2 gram omega-3 fatty acids as fish oil capsules containing 1 gram DHA and 200 milligram EPA, and at the same time we provided dietary counseling to reduce arachidonic acid intake down to 90 milligram per day, resulting in a ratio of 3:1. The control group received usual recommendations for a healthy diet, and this was equivalent to a ratio of around 7:1. Primary endpoint was the development of body fat mass in the offspring at different time points. We did skin fold thickness measurements, we did ultrasound, and at the age of 5, MRI scanning. And this is now the 1-year data on skin fold thickness, a primary endpoint, and the summary of this analysis was that there was no effect of the intervention on skin fold thickness during the first year of life. We followed up the offspring up to the age of 5 years and did repeated analysis of adipose tissue development, and at 5 years we also saw no significant influence of this change in omega-6, omega-3 fatty acid intake during pregnancy on adipose tissue development. Also, applying MRI did not show any evidence for differences in body composition, including adipose tissue distribution. So, to our more or less disappointment, there was no evidence that this intervention is helpful to prevent obesity at early age. In 2007, a new technology entered the field, genome-wide association studies looking at gene variants over the whole genome. And this is one of the most famous papers here from Tim Frayling from England. And they found and reported that an Fdo variant is a strong obesity signal. And this caused our interest in this area. And We then applied for money and defined a big project together with Thomas Illich from the Helmholtz Center Munich, and we wanted to characterize these SNPs and to elucidate the mechanism explaining these associations. I was lucky enough to recruit Milena Klausnitzer in those days as a PhD student. She had the courage and the power to really develop a completely novel bioinformatic approach, which we called PMCA, which stands for Phylogenetic Module Complexity Analysis, and use this to characterize such new data. To explain this very briefly, PMCA exploit the presence of complex patterns of evolutionary conserved and co-occurring complex transcription factor binding sites, as briefly showed here. So we did not look for sequence conservation. Instead, we looked for the functional conservation of such complex modules, and this turned out to be really an effective way to find causal gene variants here. We used the beta gamma gene locus as a showcase. This locus contains, in those days, 23 SNPs in close neighborhood, and using PMCA, we reduced the candidates to 6 and adding information on the transcriptional regulation of diabetes, we could identify a specific locus and then did a lot of biochemical analysis to get information about the mechanism. And this is summarized here. So what we found is this SNP where the transcription factor has an increased binding affinity, and this may result in a reduced beta gamma expression, which may lead to elevated free fatty acids and may cause insulin resistance in this case. We next turned to the Fdo locus, which is even more complex, and Milena tried to develop a workflow to systematically analyze these 89 variants in high LD with the tagging SNP. And as shown on the next slide, we started out with looking for the tissue which is associated with the genotype, and we found here mesenchymal stem cells. We next applied PMCA to select SNPs of interest here, and then we did a lot of validation studies as shown here, including animals in transgenic mouse models. And this led us to the following model of the Fdo locus association with obesity, and we identified this specific SNP, and the potential mechanism of action is that it leads to a de-repression of IRX3 and 5, which are master regulators in the cell, and this results in a shift towards the development of lipid storing white adipocytes, whereas the development into beige fat cells is reduced. We could also see a 5-fold reduction in mitochondrial thermogenesis in adipose tissue, and this indicates that the mechanism of action is taking place in adipose tissue itself. This is probably not the only mechanism explaining this association, but I think it's an interesting and important one. We then tried to validate this further in human studies. We were not successful when we exposed humans to cold exposure and measured increase in thermogenesis, independence of a genotype. So maybe the effect was too small to be detectable by measurement of energy expenditure. And we then moved to study metabolomic patterns in carriers and non-carriers of this specific genotype. Our first attempts were also not successful. We used targeted metabolomics, but then established a collaboration with a group from Helmholtz Munich, Philippe Schmidt-Coplin and Sara Forchisi. They have a very powerful metabolomics machinery here, and we compared circulating levels between carriers and non-carriers of this gene variant. This work is still ongoing, but what we found is really a large number of different metabolites here. They were clustered into compound classes, and most interesting to us was the class of steroid hormones, and we are trying to further elucidate what are the potential mechanisms, and this is probably a new method to also identify circulating biomarkers for such genetic effects. We are also interested to further develop new ways to characterize adipose tissue, and I would like to introduce two novel techniques which are developed right now in collaboration with different partners. One is a machine learning approach to do adipose tissue phenotyping from histology, and the other one is a non-invasive characterization of adipose tissue based on MRI. The promises here are that we can use automatic or automated approaches for more rapid and more comprehensive phenotyping of adipose tissue samples, and MRI is non-invasive, and this is also an advantage in the context of clinical studies. This machine learning approach goes back to Craig Glastonbury from the group of Cecilia Lindgren in Oxford. There was a lot of input from Milena Klausnitzer, and we provided samples and did a lot of validation work done by Julius Honecker, one of my PhD students. And the promise and the opportunity we have here is that this can probably easily expand it to other features of adipose tissue. So we are trying to annotate additional structures from such conventional HE stained adipose tissue sections, such as vessel formation, fibrosis. So there's still a lot of additional information in adipose tissue which could be exploited but by such novel and automated technologies based on machine learning. The last point is MRI scanning of adipose tissue, and we were happy to have this close collaboration established with Dimitris Karampinos from our Department of Radiology. He is an expert in MRI technology and got an ERC grant on the phenotyping of adipose tissue by MRI. And we were recently successful to establish a method to map lipid droplet size in adipose tissue using a 3 Tesla system. I will not go into detail, it's really complex, and we are trying to develop this further, but in this study we could validate this novel technique with conventional histology, and this is potentially a future thing to do adipocyte measurement in vivo without biopsies, and this could be easily repeated. This is our dream here. Another potential of this MRI technology is that we can measure the so-called proton density fat fraction, which is a kind of mixture of all components of adipose tissue. And we think that this can help us to differentiate between white and brown or beige adipose tissue. To give you one example for this, here we took a normal weight female and an obese female, and we did MRI scanning in the supraclavicular region and in the gluteal region, and we can here assess this PDFF and use it as a measure for contamination or presence of brown and beige adipocytes. So this is another possibility to better characterize and assess subtypes of adipose tissue. This was my last slide. So you see we are still active in this area, and there are really many new things which can further develop. So there is no problem to have enough challenges for the coming 40 years. And I would like to summarize some main findings here. So we have now discovered adipose tissue as a very active endocrine organ in a complex crosstalk with the microenvironment and the whole body. And this is also mediating at least some of the well-known obesity-related comorbidities. Fat cell enlargement means low-grade inflammation and insulin resistance. And we have, I think, growing evidence that genetic variants are also involved in adipose tissue function, as shown in two examples. And we have now really the perspective that novel methods are available to better phenotype adipose tissue. And this provides really big opportunities to get a better insight and to use these techniques also for clinical application at some day. I do not know when, but it's really very promising. My last part is to acknowledge the contribution of many cooperation groups and partners and friends. This is a brief list of the most important collaborators. I will not go into detail. I will also acknowledge the funding from different sources, and particular thanks to my team members, many postdocs, many PhD students. So this is really a teamwork and nothing else. And finally, last not least, greatest thanks to my family, to my wife Dagmar and our two children for giving me the freedom and support which you need to do such intensive research. Thank you for your attention.
Hans, for this great presentation. I'm amazed to see what you have done in your during your career, and I think it's really inspiring for new investigators. Yeah, now new research, new opportunities using these techniques. I have a question regarding childhood obesity and even pregnancy. With these new techniques, there is, I'm sure, opportunities to know more about the fat cells and metabolism and as well as histology. What would you have done? You started with some studies with pregnant women and also studying newborns. So do you think that these new techniques could be applied to research in pregnant women and during early childhood? And what do you think that we could eventually expect to discover that could help us to both prevent and manage childhood obesity early in life?
Thank you for this very important question. I'm fully convinced that we have to start early with our activities, and pregnancy and early childhood are the best time points for sure. I'm no longer convinced that this concept of fetal programming is really correct. So this hypothesis claims that it's the programming effect very early in life, which holds on the rest of your life. I think this is probably not really correct, at least our circumstances. We have studied this. This may be different if you have really severe starvation or something else, but not for our affluent societies, because our systems are really highly adaptive and flexible and can learn and can really adapt to changes in the environment. Nevertheless, I think that we have a big chance by advising pregnant women to look for an adequate weight gain, for example, that this may help to have an optimal fetal development. So we are also doing studies to prevent this excessive weight gain, which is a main problem today in all countries. And so far, all intervention programs were not successful because the environment and all the influence from outside is very, very strong to pregnant women. So this is really a challenge to offer better solutions for pregnant women, because they are in principle highly motivated, but it's very hard to help them and to establish this in their daily life. So this is a true challenge. So we need really societal changes. It's not a matter of molecular biology. We need really this societal support, and we are just at the beginning to get the support from outside.
Thank you very much, Hans. I really hope that you will keep going and keep researching in this field. As a pediatrician, I'm definitely convinced that we need more information, new intervention, and to fight against childhood obesity. So once again, I would like to congratulate you for receiving this Friedrich Wasserman Award. And I really wish you all the best for the next years to, I hope, continue to research and discover new knowledge for the future of the prevention and management of obesity. So thank you very much.
Thank you, and thanks to the award committee and to the whole audience.